student. I am Supriya Bankar from the Rai Shikshan Samstha's Mahatma Phule College. Today's topic is enzyme from the S.Y.B.Sc topic. So today's topic is enzyme. In this topic we have to see the basic introduction, that is definition of enzyme, definition of cofactor, coenzyme, then properties of enzyme, active site of enzyme. First we have to see the definition of enzyme. Enzymes are biocatalysts, protein in nature. They catalyze all chemical changes in living cell without being changed themselves. Next, the active form of enzyme is called as what? Holoenzyme. Holoenzyme, definition of holoenzyme. Complete active enzyme in a living cell is known as holoenzyme, which is a complex of apoenzyme and coenzyme. So basically what is meant by holoenzyme? Holoenzyme is an active form of enzyme which consists of apoenzyme and coenzyme. Next definition is apoenzyme. Apoenzyme, it is a protein part of enzyme and major part of enzyme which play important role in catalysis. Apoenzyme is a protein part of enzyme and it is a major part of enzyme. Next definition is cofactor or coenzyme. The catalysis of enzyme requires help of non-proteinic molecule which are known as coenzymes or cofactor. Three definitions. Holoenzyme is what? It is an active form of enzyme which is called as what? Complete enzyme. It consists of two parts, apoenzyme and coenzyme. Apoenzyme is a major part and which is in a protein in nature. Next is a cofactor or coenzyme. It is a non-protein part and which is required for the catalysis of enzyme. This is the diagrammatic representation of enzyme. Holoenzyme consists of apoenzyme, which is a protein part, cofactor, which is a non-protein part. Both combines gives us to what? Holoenzyme. In a diagram, see, there is a major part called as what? Apoenzyme. There is a presence of cofactor, coenzymes, and there is a cavity-like structure called as what? Catalytic site. This is the definition of cofactors. The inorganic part of enzyme is known as cofactors. Cofactor play important role in catalysis by serving as a proton donor or proton acceptor. Simply cofactor. Cofactor is what? Inorganic part of enzyme which is required for the catalysis. That can be served as a what? Proton acceptor or proton donor. When the cofactor is absent, the enzyme cannot work. So this is required for the functioning of the enzyme. This is the table. These are the example of cofactor: copper, iron, potassium. Copper required for the enzyme called as what? Cytochrome oxidase. These are the enzymes of electron transport chain. Understood? Cofactor is what? Inorganic part of enzyme and which function as what? Proton donor or acceptor. And when the cofactors are absent, enzyme cannot work. It is essential for the functioning of the enzyme. Next definition is what? Coenzymes. What is the definition of coenzymes? The organic part of cofactor is known as coenzymes. It is also a non-proteinic part. Many enzymes require coenzyme as an essential component for their catalysis. Coenzyme function as an electron-proton donor or catalyze group transfer and carry out catalysis. Bacteria and eukaryotes have ability to synthesize their own coenzyme, but higher animals need coenzyme through a dietary source. The dietary source is preformed coenzyme known as vitamins. Cofactor is a what? It is an inorganic part. Coenzyme is an organic part. Both are non-protein in structure. So coenzyme also act as what? Electron-proton donor and they can transfer the groups. In a small bacteria and some eukaryotes have ability to synthesize this coenzyme. But mammals needs take the coenzyme, that is called as a vitamins in our diet. Understood? These are the definition of cofactors and coenzyme. Next definition is what? Prosthetic group. A coenzyme or metal ion that is very tightly or even covalent enzyme protein is called as what? Prosthetic groups. Prosthetic groups means what? When it is tightly bound to the enzyme, then it is called as what? Prosthetic group. Up till now we have seen the definition of Holoenzyme. Holoenzyme is a what? It is a complex of coenzyme, cofactor and apoenzyme. Apoenzyme is a protein part. Coenzyme and cofactor are non-protein part. Cofactor is a what? It is a inorganic part. Coenzyme is a organic part. And next definition is what? Prosthetic group. Prosthetic group is a what? It is a coenzyme or metal which is tightly attached to the enzyme. It is called as a what? Prosthetic group. Some examples of coenzymes: biocycling. This transfer the carbon dioxide and dietary source is a what? Biotin. Next is a coenzyme A, which can transfer the acetyl group and dietary source is pantothenic acid and other compound, and rest of the coenzymes. Understood? This coenzyme A is required for the synthesis of fatty acid as a source of acetyl-CoA. Next point is properties of enzymes. In first we have to see physical and chemical properties. First point, large complex chemical molecule with large molecular weight. Chemically, enzymes are proteins. Many amino acids are present which result in a polarity or net charge on enzyme. Protein is nothing. Enzymes are nothing but the protein, and protein is made up of amino acid, and some amino acids have a charge, so that enzymes is having a charge, that is having a polarity. They are structurally three-dimensional with secondary, tertiary and quaternary level of protein part of enzyme. Enzymes have a three-dimensional structure. They have secondary, tertiary and quaternary level of structure. Most of enzymes are soluble in aqueous condition. However, if their polarity change in a pH and salt concentration, they can be insoluble. Most enzymes are soluble in aqueous solutions, but if there is a change in pH or change in salt concentration, they are insoluble. They are sensitive to changing pH, temperature and salt. Obviously, enzymes are sensitive in pH, temperature and salt. They work in optimum pH, temperature and salt concentration. Their electric charge allows them to move in an electric fields. So, in case of electric field, when we apply the electric field, protein or enzyme can move. Enzymes can be precipitated with acids, alkali, salts. So, this property can be used for the enzyme purification. Next, catalytic properties. Enzymes conduct the chemical reaction in living cell at faster rate, normal atmospheric pressure, normal pH, normal temperature and normal salt concentration. Enzymes conduct chemical reaction at a faster rate and normal condition, that is normal atmospheric pressure, normal pH, normal temperature and salt concentration. Enzymes increase the rate velocity of the chemical reaction. When we compare the two reactions, that is catalyzed reaction and uncatalyzed reaction. Catalyzed reaction means what? When the enzyme is present. Uncatalyzed reaction means what? When the enzyme is absent. When we compare the rate of reaction, what is meant by rate of reaction? The number of reactant getting converted into product in a per unit time. For example, 10 moles of substrate is getting converted into product per unit time. This is called as what? Rate of reaction. Catalyzed reaction have more rate than uncatalyzed reaction. Enzyme conduct chemical changes at low activation energy. So what is meant by activation energy? Energy required to change the conditions, to change the temperature, pH. But enzymes conduct a chemical change at a normal conditions. So catalyzed reaction require low activation energy than a uncatalyzed reaction. Understood? During catalysis, chemical change take place by one of the following mechanism. acid-base catalysis, covalent catalysis and metal ion catalysis. In upcoming video we seen these mechanisms. This is the graphical representation. In a first graph, this is an uncatalyzed reaction that require a high activation energy. Why it required high activation energy? Because energy required to change the conditions, to change temperature, pressure. In a second graph, this is graph for the catalyzed reaction, and there is what requirement of low activation energy. Why? Because in a catalyzed reaction, the reaction occurred at normal temperature, pressure. Next property is specificity. What is meant by specificity? It is a property of an enzyme to bind to the substrate known as a binding property. It is a selective and specific binding. There are three types of specificity. First one is a what? Absolute specificity. Second is a stereo specificity. And third is a broad specificity. First, absolute specificity. Specific enzyme bind to specific substrate like a lock and key. Example, ureas act on a urea. So, specific enzyme can bind to only a specific substrate like a lock and key. Key can lock and key. Example is what? Ureas acts on a urea. Second type of specificity is stereo specificity. Some enzymes are highly specific and identify stereoisomers, that is L and D form. Example, L-amino acid oxidase acts on L-amino acids only. D-amino acid oxidase acts only D-amino acids. Next type is what? Broad specificity. Enzymes acts on more than one structurally related substrate. Example, hexokinase catalyze phosphorylation of glucose, fructose and mannose. What is meant by broad specificity? Enzyme can act on the structurally related substrate. This glucose, fructose, mannose, these three sugars have similar structure. So, hexokinase can act on the glucose, fructose and mannose. This is the specificity. Three types of specificity. Absolute specificity, that is specific enzyme can bind to only a specific enzyme. Stereo specificity, they can even identify the stereoisomers, that is L or D form. Next, broad specificity, enzyme can act on the structurally related substrate. Next, these are the properties of enzyme. Next is a active site of an enzyme. So, what is meant by active site of enzyme? The active site of an enzyme is the region that binds the substrate and convert it into the product. So, on the active site, substrate is binding. It is a small part or cleft on enzyme which is responsible for substrate binding, specificity and catalysis. Actually, catalysis is occur on what? Active site of an enzyme. It has shape, size and charge functional group that decides substrate binding and specificity. Active site is having a particular shape, size and having a charge because of presence of charged amino acids. The side chain of amino acids project into the pocket of active site and serve as a charge end for substrate binding. It is the tertiary folding of enzyme which allows formation of cleft pocket which decides specificity towards the substrate. The charge functional groups of side chains are responsible for substrate binding and catalysis. Serine, cysteine, lysine, histidine, tyrosine, these are the commonly occurring amino acids at the active site. Basically, what is meant by active site? It is a pocket-like or cleft-like structure present on the enzyme where substrate can bind and where actually catalysis can occur. It is having a charge where the charged amino acid present. Understood? In next, this is the structure of active site. This is the Holoenzyme. And this red color is what? Catalytic site and binding site. This is the structure